Hi everybody, welcome back. We are continuing topic 5.3 on Mendelian genetics today in our 5th unit on heredity. In our last video, we got an introduction to what genetics is all about, what it's used for, and we discussed Mendel's 4 concepts when it comes to his model of inheritance, is that there's alternative versions of genes called alleles, and each organism has 2 alleles that it gets one from each parent. And gametes, when they fuse, they only have one allele, one alternative version of a gene, so that when they combine, each organism has 2 alleles, and those alleles determine the trait. More specifically, if there's a dominant allele present, it will be expressed over what we call the recessive allele, which will be masked by the dominant allele. All right? So let's get into how we can use genetics and this knowledge of that we got from Gregor Mendel in order to predict what we can see in subsequent generations. And these Punnett squares here and dihybrid crosses, these are going to be illustrated pretty well they're going to illustrate Mendel's concept of inheritance pretty well here, as we'll see in just a moment. If you're familiar with these, great. Just keep watching though. You know, it's going to still be pretty helpful. All right, so here what we have over here is a Punnett square, and it's a tool for using predicting allele composition of offspring from a cross with known genetic makeup. Okay, so this is the example that we brought up before. This is our P generation, and not as in like P as in pea plants, or I mean P as in parental generation. We've got a true breeding purple flowered pea plant over here, and we have a true breeding white flower pea plant over here. All right, and if we cross them, meaning we take the genetic information from this one and we combine it with the genetic information from this one, we can use this to predict what the ratios of the next generation will be. All right, and if you remember from the last video, the F1 generation, when we crossed the purple true breeding plant and the white true breeding plant, they all were purple still in that F1 generation. Spoiler alert, sorry. But another term that I'd like to introduce to you here before we get going is that these plants are what we call homozygous, which means they have identical alleles for a gene. All right, as you can see, each organism, according to one of Mendel's concepts here, has 2 copies of a gene, right? So both of its copies are what we call the dominant allele up here, that's homozygous. Both of its copies are the recessive allele down here. So we can say this flower is homozygous recessive and this flower is homozygous dominant. All right, so remember, what happens when we cross these? Well, we get all purple flowers, right? But why do we get all purple flowers? Well, because the dominant allele, this is another one of Mendel's concepts, the dominant allele is expressed over the recessive allele. So that means that the purple trait will be expressed over the white trait. All right, but check out how we use this here. We can I'm going to go back to the last page so I can show you. What we do here in order to solve this Punnett square, okay, we take this allele, we take our pairs of alleles and we kind of orient them around the square here, okay, and then we bring this allele all the way down here and this allele all the way down there, and then this allele, and we write these across, okay, and then as a result, as we can see, we got 2 alleles per box. This allele's going to be in here. I'm just going to show you the next page instead of me writing that out. Okay, as we can see, there's a dominant allele here, dominant allele there, dominant allele here, dominant allele there, recessive allele there, so on and so forth. Okay, so what we end up with are all what we call heterozygous offspring. So in our F1 generation, we end up with all heterozygotes, and that means they have 2 different alleles for a gene. Okay, remember, 2 copies of a gene. All right, one of these alleles is dominant, one of these alleles is recessive, therefore it is heterozygous. Hetero means different, homo means same. Okay, what we're looking at here, all these representations of letters are what we call genotypes. Okay, so you can have a homozygous or heterozygous genotype, and the genotype really determines what we call the phenotype, which are the observable traits like the purple or white flowers. Okay, so a homozygous dominant phenotype, or excuse me, homozygous dominant genotype will produce a purple flower, which is the phenotype, right? A homozygous recessive genotype is going to produce a recessive phenotype, which is the white flower. Okay, and then heterozygous, you know, if there's one dominant allele, that's how it's going to go. It's going to dominate the other one, right? A heterozygous genotype will make a purple phenotype. Okay, so the bunch of terms that you may have heard before if you've taken a biology class, or if you had this in like 7th grade or something. Great. All right, let's keep going. So those are our F1 generations, okay, that we cross those. We have a bunch of heterozygotes, right? And if we cross them together, okay, with what we call a monohybrid cross, we're going to get something different. Okay, maybe if you remember from the last video what the ratio was of purple to white flowers. In our last video, I talked about what the ratios ended up being. But we can solve it with this Punnett square over here by doing what we call a monohybrid cross. These are both hybrids, right? So if we do this Punnett square, okay, we're going to have a dominance allele there, dominance allele there. Done. Okay? And then we're going to have a recessive allele here and a recessive allele there. See how I brought this down and brought this down? Okay, and then if we go across, we have a dominant allele there. We have a dominant allele there. We have a recessive allele here and a recessive allele there. All right, so I successfully did this this Punnett square. Okay, now we can determine, based on our knowledge of dominant and recessive phenotypes and genotypes when it comes to these flowers, okay, we can determine what the ratio of purple to white flowers is going to be. Remember, a homozygous dominant phenotype is going to produce purple. Homozygous recessive is going to produce a white flower, and then these heterozygotes, take a look, they're going to produce purple flowers too. Okay, so what's our ratio that we end up with for purple to white flowers? We get a 3-to-1 ratio. And you probably remember that from the last video, but that's okay. Crossing two heterozygotes in a monohybrid cross produces a 3-to-1 ratio. Okay? So that's how you do a Punnett square. This is what we call a monohybrid cross. But what happens if we take it a step further and we do a dihybrid cross? Well, remember that law of segregation that, each gamete is only going to get one type of allele, okay, and they're going to randomly segregate independent of all the other ones. Well, we can figure that out. We can see that from the result of a dihybrid cross. Okay, so let's get into two more pea plant traits here. I'm going to introduce you to another experiment that Mendel did. All right, he crossed a true breeding pea plant that produced yellow, smooth peas. Okay, yellow smooth, that's represented by this dot over here. And he crossed that with a true breeding green pea or green wrinkled pea plant. Okay, so here's our genotypes and phenotypes down here that I've listed for us. Okay, if you're homozygous dominant or you're heterozygous for pea color, okay, you're going to have yellow peas, and if you're homozygous recessive, you're going to have green peas. Okay, something similar down here. If you have homozygous dominance or heterozygous for pea smoothness, okay, you're going to have smooth peas. All right, and if you are homozygous recessive, you're going to have wrinkled peas. Okay, so this is our double recessive guy over here, and we're going to cross it with our double dominance over here. All right, so if we cross both of these, if we cross these plants for both of these traits, our F1 generation is going to look very, very similar to what we saw in our last experiment. It's going to be all yellow smooth peas, right? Because if we cross all these, no matter what, okay, we're going to end up with heterozygous. So one dominant, one recessive allele for both. Okay, so here's our F1 generation. But what happens when we cross these cross-pollinate the F1 generation? I can't talk right now. Okay, so here's the question I have for you. Do the hybrids transmit the same allele combinations as the parents? Okay, and in order to get this, it really, really would help to show you on a dihybrid cross, because it's kind of like, I don't know, it's kind of hard to visualize. All right, so take a look at this. If we don't follow what we call the law of independent assortment, okay, then once we cross all of our F1 hybrids, okay, we're going to end up with our 3-to-1 ratio, just like we did with the monohybrid cross. Okay, so if the dominant alleles segregate together and the recessive alleles segregate together, then we will end up with that 3-to-1 ratio. Okay, that's what this law of segregation means, is that it's completely random which alleles will go into which gametes. Okay, so here's the deal. This is not what we actually got. This is not what Mendel actually got when he crossed these two plants together. This is not the ratio that we ended up with. Okay, this would be the case if dominant alleles only segregated with dominant alleles and recessive alleles only segregated with recessive alleles. Instead of that 3-to-1 ratio, we got a 9-to-3 to 3-to-1 ratio. What? You'll see. Okay, so Mendel was expecting that 3-to-1 ratio, the yellow smooth and green wrinkly 3-to-1 ratio. Okay, but he also got some that were yellow and wrinkly and green and smooth. Combinations of both. You got more combinations than what we had before. And how is that illustrated? Well, that's done through a dihybrid cross. Okay, now this is the same thing as what we did before in the monohybrid cross, except it's for two traits, right? So check it out. Remember, our F1 generation, here was our genotype. We had one heterozygous and another heterozygous. Okay, now check it out. From this genotype, okay, we can get 4 different possible combinations of gametes. Okay, we could get one where they have both dominant. They could have one dominant and one recessive, one recessive, one dominant, and both recessive. Okay, you got all these different combinations of gametes that you could get from this parent. Okay, and you're going to get the same thing on the other side. All right, you could find these out, which gametes, through the FOIL method, which I will show you later on in class. Promise. Okay, if you need extra help with this, I can make another video, but I was not ready for it right now. Okay, but what I'm trying to say here is that the dominant alleles don't have to go with the dominant alleles, and the recessive alleles don't always have to go with the recessive alleles. You can get different combinations of the dominance and recessive for 2 traits or with these gametes over here. All right, so if you cross it, I'm not going to walk through this whole thing right now, okay? But as you can see, we end up with 1, 2, 3, 4, 5, 6, 7, 8, 9 yellow smooth peas. We end up with 1, 2, 3 yellow wrinkled peas, 1, 2, 3 green smooth peas, and 1 green wrinkled pea. Okay, and you're doing the same thing as before. You're just dragging these alleles down. Okay, and then you're dragging these alleles across, and you get a new combination of alleles in each one of these boxes. All right, and each one of these is going to produce a different genotype, different phenotype. All right, so what I'm trying to say here is that dihybrid crosses indicate the law of independent assortment, that each pair of alleles segregates independently of any other pair during gamete formation. That's going to be important to know. Okay, so Mendel's law is what we've been talking about in this whole thing with Punnett squares and dihybrid crosses. These apply to genes that appear on different chromosomes than one another. So in our next video, we're going to talk about when Mendel's rules don't work, and then we'll get into what we call chromosomal inheritance as well. Okay, because sometimes genes are on the same chromosome, and that causes a whole other different thing. Okay, and I believe, yep, that's it for this video. Please let me know if you have any questions, and we will see you next time.